I'm Meredith Blackwell, and I'm here to conduct an interview with my colleague Don Pfister. Say hi, Don.
Hi.
And we're interviewing David Geiser.
Hi.
And David is going to tell us something about his life with fungi. We are at the Mycological Society of America meeting in the year 2019 at the University of Minnesota in Minneapolis, Minnesota. So let's ask David some questions.
Okay.
Don?
Where were you born? What's the beginning here?
I was born in Orange, California. In
Orange County or just
Orange?
Orange?
No, the city of Orange.
Oh, okay.
In Orange County. Back when there was still agriculture in Orange County, California. So, you know, at the end of the road there was an orange grove. That's no longer true. And in fact, about 15 years after I was born, it wasn't true anymore.
And your parents were?
So my father was a salesman of various sorts, but for the last 25 years of his life, he had a security guard business. So he put security guards in buildings, mostly in Los Angeles. Before that, he did a lot of different things. He was actually, when I was born, he was a probation officer for Orange County. And my mother was a homemaker, but she, for years before she was married, worked for A.B. Dick Company and she demonstrated mimeograph machines in Chicago.
Neat.
Yeah, nobody knows what they are
Yeah,
anymore.
exactly.
Mimeograph machine.
And my mom could take one apart and put it back together.
Forgotten
Certain skill.
Yeah, exactly.
Yeah. So had they been to college or?
My father did. He did GI Bill. He was.
Yeah.
He went into the Army right after World War II ended, and was actually sent to Korea, but before the actual Korean War started. But he was there for 3 years. But he got GI Bill and went to Creighton University in Omaha.
Yeah.
But my mother didn't. She started working at age 16, and when World War II started, and worked until 1960 when she got married.
Well, it was a question. Well, it didn't. It just came up with a number of people who mentioned they were the first ones in their family to have gone to college, and that includes a lot of prominent mycologists.
Yep.
Yeah.
So,
Yeah, I wasn't the first in my immediate family. I've got 3 brothers and 1 sister.
Wow.
My 2 older brothers didn't actually graduate from high school. And that was when my parents decided, well, it's Catholic schools for the rest of you. And then my older sister, myself obviously, and my younger brother, we all completed college degrees.
great. So do you have some strict Jesuits or something?
I had Servites,
Oh, okay.
which were not, kind of average in that regard.
So when did you first get interested in fungi, or do you remember?
It really was in graduate school.
Okay, well, let's go back to undergrad school then.
Yeah.
We'll get the first fungus later.
Well, I got a degree in ecology and evolutionary biology at Northwestern.
Okay.
And
In Illinois?
in Illinois, Evanston, Illinois.
Yeah.
And actually, one of my favorite courses, though, was the diversity course. And it wasn't so much, the animals and the plants that were interesting. It was all the weird stuff. And I mean, weird stuff being anything. I guess that's not an animal or a plant. And so that kind of carried on. But I really, it was really in grad school.
And so you went to grad school not knowing what you were doing? How did you focus then?
Well, so my degree's in genetics, and it was, and at University of Georgia, the genetics department was almost split 50-50, evolutionary biologists and people doing molecular genetics and whatnot. And so it wasn't really organismally focused, though, necessarily. I was going to work on Drosophila, chromosomal inversions and things like that. And but then we had a rotation system the first year, and the last rotation that I did was on fungi, and I liked the lab. And
was it Bill Timberlake?
It was Bill Timberlake, right? It was kind of funny how that happened. I was really kind of planning on working with Wyatt Anderson, who's a very well-known evolutionary geneticist. And
he's a National Academy member.
he's a National Academy member. And, but I don't know, I think he thought that I needed maybe a little bit of I don't know, a kick in the butt or something. I don't know what it was, but he basically made it clear to me that he wanted me to do a rotation in Bill's lab. And I think because he knew that that was a very, it was a disciplined lab.
Oh.
And Bill had, of course, a reputation among students as being really tough. And so I did it, and best decision ever made.
That's funny. I wouldn't have thought of him as being very disciplined.
Okay, interesting. Yeah.
I remember when
Well,
he gave a talk. He was an invited annual lecturer, when we called it that, for MSA.
Mm-hmm.
And he got up and told us he had spent his MSA, I guess it was his fellowship money.
Yeah, it was MSA.
Spent it on a top-notch audio system.
Yeah, he told me that too.
Oh, he did.
And now everybody knows.
Yeah, and I remember the talk. He had little circles, and they were filled in. And anyway, I didn't get the idea, but the sun was going down, and so when we got to the end of his talk, it was the little semicircle was all black.
Oh, okay.
It's kind of cute.
Yeah, yeah. But it was a
It was a good lab for you.
Yeah, exactly.
You learned discipline.
Well, I mean, I think I actually had some discipline up to that point, but it was, you know, culturally, it was a very different kind of lab than Wyatt's lab. Mm-hmm. And it was a much more, Bill's lab, people were very active in terms of criticizing one another's work. Oh. And so there was, in a positive way. Mm-hmm. I mean, you were always, people were asking you what you were doing and kind of challenging you. And so I kind of, I liked that. Yeah, so.
I remember the first time I met you, and it was actually, you were in the program it was an NSF program?
NIH, yeah.
NIH, okay.
Yeah,
and Charles Mims and Timberlake and the biochemist.
Yeah,
there were a number
Claiborne
of,
Glover, and so it was a NIH training grant,
right.
Not a real big one. I think it supported 4 or 5 students at a time. I think it was molecular and cellular biology of fungi. Mm-hmm. That Charles and Bill wrote, because you know they collaborated. They had a really nice project together, that benefited. You know, there are a number of people. Karen Snetzler was supported on that grant. Somebody who's been very active in MSA. Mm-hmm. Myself, Mary Stringer, who's worked now for Novozymes for 20-some years. Can't think of other people off the top of my head, but yeah.
Yeah, I can remember Bill and Charles were doing spore development, or was it aspergillus?
Yeah, aspergillus conidiaphore development.
And they got a cover picture of on a plant name journal. What was it?
Plant Cell.
Plant Cell, yeah.
Yeah, and I think some others too, maybe. You know, and you know, I think that's some of the best work that came out of that project was the work that Tommy Sewell was the electron microscopist.
Oh yeah, I remember him.
And working with Charles, and just beautiful characterizations of these developmental mutants in aspergillus. Mm-hmm. And you know, I see those pictures now all the time. Those are the electron micrographs of aspergillus.
I just thought it was neat. It was on a plant journal.
Yeah. Well, yeah, that's right.
Okay, so you finished up your PhD and went west again.
And went west again. That's right, to Berkeley, and worked with John Taylor, which was, you know, I'd known John and everybody. Well, maybe not everybody in his lab, but they're so visible in the MSA community in particular. So going. I really knew exactly what I was getting myself into at that point. Which was great.
Yeah, and what did you do there?
Well, I worked on Aspergillus flavus, and you know, John. There was a lot of. We've talked about that a lot in this meeting. How John kind of gave people freedom, and you know, we really didn't have funding at that was specific for that, but he had money for a postdoc really I was supposed to be working on cocci, I think. And I did a little bit of, I tried to isolate it from soil, which never did work out. Turns out that remains a very difficult thing to do if you can get it, if you can accomplish it at all.
Blasto and cocci both, I think.
Yeah, that's right.
Because some of those things are awful.
Which is an interesting problem, that they're clearly soil saprophytes, and yet you can't isolate them.
With blasto in Louisiana, you take a dog and put him in a spot, and he gets sick.
Oh, I see. Well,
yeah. Well, not that anybody does it experimentally,
right,
but it's
but
the way it happens. And you
bio
go to that spot.
bioassay.
You go to that spot, and you can't get it.
Yeah, I was worried that I was going to get it, and because I was collecting.
I meant that
you
couldn't collect it.
Right. Yeah. Exactly. I drove. I don't remember how many soil samples I collected in the garden spots of California, like Bakersfield, Blythe, where cocci tends to be, and didn't find any. I did get a rip-roaring respiratory infection though.
And you thought
Right afterward, and I thought, oh no, but I had it cleared, and then not too long later, an MD came by and gave us skin tests, and I was negative.
So you didn't have it.
Apparently, I did not have it.
It was all psychological.
Yeah,
Well,
right.
Yeah.
So,
Good.
yeah, nobody else in the lab got it either, to my knowledge.
Yeah.
So then,
I got a job at Penn State in 1998.
and at the beginning, were you in charge of the fusarium operation?
Yes. So the fusarium operation being the culture collection, that Paul Nelson had started.
Had Nelson died or
Nelson
retired?
died. Yeah. So Nelson died in '96, and then I got the job in '98. It was interesting. The job had been advertised, for a I think it was systematic mycologist, and it listed, there was something about fusarium in the job description, but there's also something about aspergillus and maybe even penicillium. And I wrote my aspergillus job prospectus and sent that, and it took quite some time for them to come back to me and say, we're interested, but we really want somebody who's going to work on fusarium, and I had to sort of prove that I was willing to do that. But it's just, if that's what they wanted, and I thought about it, I thought, well, I can do everything I am interested in doing in aspergillus more or less on fusarium. So, didn't really matter to me.
And it worked out well.
It worked out very well, yeah. And I had no idea what even the fusarium community was. The person that I knew who was the best in the community was Carrie O'Donnell at that point.
Sure.
And one of the questions that I had about the job is, are you really sure you want to hire somebody to do molecular systematics of fusarium? Because there was somebody who was very much on top of that.
Right.
And so I, and I went to Carrie and talked to him about it at length. And what he said was that, oh, of course, I'd love it if you got that job. That would be great. And it was. I mean, and Carrie's, of course, an amazingly generous collaborator, and and just such a strong scientist with really high standards that it's been terrific working with him. And it was good. I think it was good for Carrie as well, because he was sort of a voice in the wilderness prior to that. And I mean, it helped, of course, that he was publishing these, at that point, really, in the late 90s, his really hot papers were coming out where he was demonstrating very clearly what the real story was with fusarium taxonomy. Yeah. But I think it helped him to have somebody else who was saying the same kinds of stories.
Lots of species.
Lots of species, and because it was a very, the fusarium community outside of Carrie is also really, really great, but there was a little bit of circling of the wagons that was going on prior to that, and including, frankly, Paul Nelson was one of the wagon circlers. I know that, and which is too bad, but that's the way things ebb and flow in science quite often, is people see the new thing coming along and they're not necessarily sure that that it's the real thing.
Yeah.
And they don't want to be excluded.
And they don't want to be excluded, and certainly, and they see somebody who knows a lot less than they do about certain things, and it's just a natural phenomenon. But these things tend to just.
Isn't this part of the era too, where thinking about fungi as they can go anywhere, you know, these broad distribution patterns
Sure.
and so forth, and then Carrie is kind of pushing back at this. Look, you know, we've got continental endemism, we've got other things going on here.
Yeah, absolutely. And interestingly, and you know, I don't know if you've asked Carrie, I think you've interviewed Carrie, but I'm not sure if you asked about that, but some of that didn't really pan out with further data, but certainly there is a lot of endemism, and a lot of species that have been lumped together previously do show those continental, or even on a much smaller scale, patterns. A lot of things originally in Fusarium were described. It's a Fusarium. It's on a new host.
Yeah.
You know, it's Fusarium host, and like for a lot of fungi, of course, and that was thrown away in the early 20th century for the most part, but a lot of that's coming
It's
back.
come back.
You mean the Snyder and
Yeah.
Hansen?
Right, the Snyder and Hansen
Yeah.
lumping.
Yeah.
And I mean, I was actually kind of fortunate, though, that I walked into it with none of the baggage of the past, and naive, really. That's a good way sometimes to walk into a difficult situation where you don't have any friends or enemies, you're not tied to one thing or another. I could just sort of move.
So does dispersal have anything to do, I'm sure, with the way that they're whether they're very endemic or not?
Certainly, and it's everything from, you know, probably the wind to you know, movement of plant material.
Can you generalize?
It depends on a lot of things. So, I mean, it was just announced yesterday, and I think confirmed that tropical race 4 of Fusarium oxysporum, on the banana pathogen, is in Colombia, which is a disaster. Now it's in the now it's on in the Americas. I mean, hopefully they can contain it.
Was that a paper here?
No, it's in the
it was in
news.
the news.
So it was confirmed in Holland, apparently, where they should, where I think they can do it.
So we had the Grand Michel, and then what's the current
The
banana?
Cavendish.
Cavendish, and that's the one.
And that's it's
the one in Colombia.
Right. Well, right. The fungus is
Yeah,
there. I don't know
yeah.
in terms of what what damage it's caused.
But they're
at
growing
this
that
point.
banana, the Cavendish.
Absolutely.
Everybody is.
That's
Everybody
what everybody
is, yeah.
And so, but anyway, how did it get there? Well, it could have gotten there in any number of ways, but it could have been
Hurricane?
It could have been, well, hurricanes do, right? Move things. Who knows? But it could have been on somebody's shoe. In other cases, you know, one of the things that has been heavily studied is Fusarium graminearum, which causes head scab of wheat and barley. And we're moving these commodities all over the world. You know, I've studied the ambrosia beetle, Fusaria, that are causing a lot of damage, and those move on wood packing materials, and it's a lot of the same things that you see in other plant pathogens.
So one thing that really hit the news was the contact lens problem. Can you talk any about that or not?
Oh, sure. No, I can talk about it. I think the statute of limitations is up. So that was very interesting, and it was really lucky, and in a lot of ways, because of Ning Zhang, who was Meredith's student and was my postdoc, and we had submitted a paper. Actually, it had been reviewed, and we were just going through the revisions when that all happened, and that was extremely lucky that was the case on the human pathogenic Fusarium species, including the eye pathogens. This was in early 2005, and at the same time, these reports were coming out of Southeast Asia that there was an outbreak of mycotic keratitis, so cornea infections associated with one of Bausch & Lomb's contact lens solutions. And then there were cases in the United States that were starting to come up. So in early 2006, actually is when this happened, I think I said 2005, when I started to get this, Carrie had said something to me about that there's this outbreak, and then I got a phone call from Bausch and Lomb, and they sent somebody in a car down from Rochester, New York, down to State College to give me cultures to look at. And so I immediately did a DNA prep, ran a couple of our markers, elongation factor 1 alpha and the ribosomal genes, and found that they only gave me 2 cultures at that point, but they were 2 different species within the Solani complex. We called them at that point FSSC 1 and 2. We now know them as Fusarium petroliphyllum and Fusarium keratoplasticum.
They were undescribed at the time?
They were undescribed, right. They were just all Solani at that point.
Yeah.
So I was able to tell them 2 different things, which in a sense was good news because it was unlikely that there was a central product contamination that explained these 2 cases that were in 2 different states. I don't remember which states. They were New Jersey and another state. However, the cases were very strongly correlated with their #1 selling product, which was Renew contact lens solution with Moisture Lock. You can still buy Renew. They no longer have the Moisture Lock formulation.
Was that lipid, some kind of lipid?
Yeah, well, yeah, it's a proprietary compound, I guess, or material, but it also had a different antimicrobial compound in it that they didn't use in contact lens solutions very much. And it turned out, so then there were hundreds of cases in North America and Southeast Asia and a few other parts of the world, but mostly in Southeast Asia and North America, that we found quite a few species, including Oxysporum. But they were sort of the usual suspects in terms of the Fusarium species that we had known before because of Ning's work. We'd already characterized them, so we were finding quite a few taxa, but they were the things that, and 1 and 2, petroliphyllum and keratoplasticum, are really commonly found in plumbing systems. In fact, that was all that we knew keratoplasticum from, was from highly anthropogenically influenced, either from a person, and not just eye infections, but deep mycoses, people, in immunocompromised people, or from a plumbing system, or something odd like fouling the lubrication oil in an automobile parts manufacturer. Really sort of extreme environments. Petroliphyllum loves petroleum. That came from oil, originally that name from oil, contaminated water. So anyway, it was because we were very lucky that we had done that study, and were ready to. We knew exactly, and we'd seen these fungi before, and it was more than one. Didn't explain why their #1 product, though, was the only factor that these patients had in common. Well, there weren't very many cases in Europe.
And they didn't sell it there, or?
They sold it there, but that was all coming from a different manufacturing plant. So there's something, but it wasn't that. It probably wasn't. It wasn't. There's no reason to think it was in the product as a contaminant. You were getting it in your own sink.
I remember a funny thing about that paper. Well, Ning gave some of it, at least at a meeting, and someone asked at the end, "How do you avoid getting these fusariums?" And she said, "Don't eat lettuce." So apparently some of
cucumbers, maybe.
Cucumbers, some vegetable.
Because petroliphyllum is also associated with it's a fairly weak pathogen of cucumber.
Okay.
Yeah,
but I thought it was very funny.
Yeah, that was all we had, at that point. We didn't have.
So that was before you had the one from the contact lens.
I think it probably was.
Yeah, it must have been because the paper probably wasn't out at that point.
And another thing that's interesting, and this is again, valuable information that's come out of it. So in Ning's paper, we talked about eye infections, but what we stressed was a different species than those 2. It was Fusarium, what we now call falciforme. There we called it Fusarium. Actually, we thought it was 2 things, 3 and 4. But that mostly occurs in soil. And if you're unlucky enough to get a corneal infection caused by Fusarium in southern India, where this happens a lot, the way that you're getting it, you're an agricultural worker, and soil gets in your eye. And there, it's usually falciforme, and that was most of what we had for that paper, that for Ning's paper, were isolates from agricultural. So we reported there that falciforme is disproportionately associated with with eye infections, not what we came to know as petrolophyllum and keratoplasticum, because these these contact lens infections are extremely rare.
Yeah, but without contact lens, you don't find them.
Yeah, and the problem was that that is also very important to note is that you probably, well, almost certainly had to misuse the contact lens for this to happen. And so if you reuse the contact lens solution over and over, which is, I think, the #1 lack of compliance that they experience with it, you get a buildup of an actual biofilm in the contact lens case, and then you're sticking it right on the eye.
Right.
So yeah, it's pretty awful. Yeah.
Well, you know, when I did a postdoc with Jim Kimbrough in Florida, he often had fungi to identify that were infections from citrus workers picking the citrus. And I don't even know what they were, but I just wonder maybe they were some of the
Well,
Fusarium.
so it could be.
They were corneal infections.
Well, and so if almost all of the mycotic keratitis cases in the United States are from Florida, and a lot of it is agricultural. There's an eye institute in Miami, Bascom Palmer, I think, and they processed a lot of samples there. Yeah. So I've got that was one of the nice things I had was quite a good collection of clinical isolates that Paul Nelson had assembled. I mean, he was, and did a fair amount of work on them as well. You know, we could say that it was Solanae or Oxysporum, and that was what allowed us to discuss it in the paper. So if you're a citrus worker, though, you're going to get falciforme, most likely, and again, it's probably going to be coming from soil. But there were other species as well, including some plant pathogens that people would get. Yeah, the human pathogens are interesting in Fusarium, and if you're unlikely to get an invasive infection, you know, they're very resistant to antifungal drugs. So, and they might not know it's Fusarium. So the typical dose that they would give you isn't enough. So there's particularly high mortality.
So they're a monophyletic group, the human pathogens?
No, it's what's kind of interesting about them is that there's some kind of convergent evolution. So we've got these species in the Solanae complex, which explains most of the infections. But in Oxysporum, which is in a different major clade within Fusarium, you also isolate it from plumbing systems. And it's also, though, strongly associated with, if you get an Oxysporum infection, it's probably going to be this one, what we call sequence type 33. That also, I've got an isolate of it that from paint, somebody painted their wall, and a lawn of Fusarium Oxysporum sequence type 33 came out. Yeah.
What are they eating in the paint? Are they they're getting?
Yeah, I mean, usually there's an antimicrobial compound in the paint, and it must have just, you know, not been added or it failed. But who knows? I mean, that's the other question is what are they eating? You know, Chris Smith, who is here and graduated last year, worked some on these, and we wanted to do more work on, you know, how they form biofilms and kind of understand, you know, what they do. I'm still interested in doing it, but you know, you put them in, they grow very well in just tap water, and they're making a living in plumbing, you know, and set.
So you're responsible for this culture collection.
Yeah.
How many are there?
How many? 20,000 isolates.
Yeah.
Yeah, responsible is a good way to put it because you know, I don't have any technical support for it. So it's.
How are they preserved?
Mostly on lyophilized, so which is a good, stable, you know, low maintenance way to do it. I mean, they're in freezers, which you know, the important thing is to keep them not subjected to a lot of temperature changes. And then some I keep on liquid nitrogen. Some species or groups of species don't seem to do so well in lyophilization for reasons we don't really understand. But yeah, 20,000. And you know, when I started, it was 14,000 and some, and we just don't have room for more. I mean, I've got more, but and we're going to have to do something about that. But yeah.
What about sex and these things? Or
Oh, I mean, it's like.
do you ever?
Do you see the sexual
Yeah.
stages?
Mm-hmm.
No, not very often. I mean, so if they're homophalic,
Mm-hmm.
you're quite likely to in the field.
Yeah.
So Fusarium graminearum is homophalic, but the rest of them, you know, they're mostly heterophalic. So you only encounter the sexual stage if you mate them in the lab. I think they're all probably sexual, including the things that, like Oxysporum, nobody's seen a sexual stage, but there's a lot of circumstantial evidence, including the presence of both mating types, and that to indicate that they're probably sexual as well. Just someday we'll get the sexual stage.
But the ones that you get out of infections in humans, they're
Oh.
usually.
Actually, you know, it's a good question. We've never really looked at that in terms of what the mating types are in the human pathogens. Yeah, that's a good question. You know, and I think it makes me think of Jun Quan Chung. You know,
Yeah.
who found the first
After years.
heterophalic
Cryptic, or
Aspergillus well, that too.
okay,
That's
yeah.
that's like the
Yeah.
most famous example, Phyllobacidiella. But with Aspergillus, she found, you know, from two eyes, I think, on a rabbit. One was one mating type, one was the other, and she
In
put
the
them
same
together in a
rabbit?
plate. Yeah, I think so, and got what we would call the Neosartoria sexual stage. So that was Neosartoria fennellii, named after Dorothy Fennell.
Uh-huh.
Yeah.
Yeah.
Yeah. I just had correspondence recently with.
Yeah, she's still. I mean, she's retired, but she's still
she's
quite
still
active.
working. Yeah. And she was telling me she has a daughter who's a concert pianist. Did you know that?
I did. Yeah.
Yeah, and I think she teaches too now. But she was down in South Carolina performing, and
Oh, good.
it was a night when it was really stormy, and in fact my electricity went out, so I couldn't even watch the program. It was recorded,
Oh, that's great.
but I couldn't watch it at the time.
Yeah, she's in really. We kind of had a common interest in that. You know, she was Raper's student, Kenneth
Right.
Raper's student, and has encyclopedic knowledge of Aspergillus diversity. So I talked to her quite a bit, and every once in a while, it's been a while, but she would call me, and she's very direct on the phone. You know, she's got
direct.
She's got certain things she wants to know from you, and she asks those questions, and it's very organized, one thing after the other, and then okay, good talking to you. Bye.
Yeah.
Yeah, this was she used to call me for advice on what what went where.
Yeah,
You know, she was
right.
interested in phylogeny, but that knowledge is beyond me now, and she's wanted more detailed information. So maybe she's calling you.
Well, yeah, it's been. I think the last time I saw her was in Holland. It wasn't that long ago, just a couple of years ago, I guess.
But I remember she gave a talk. She was a distinguished lecturer one year. Talked about Cryptococcus gattii.
Okay.
And
yeah.
for weeks afterwards, I was trying to figure out why that thing would be in the localities where it was.
Do we know?
Well,
at this point, it's crazy.
I think Joe
Geiser.
Heitman maybe can give us more information, but
Yeah.
I think it's got to do, he thinks some of them are associated with certain trees.
Oh, yeah, right.
And I guess the early idea was it was with some eucalyptus.
Right.
Yeah.
But something different in Canada. Yeah.
Yeah. So anyway.
Yeah.
So anything else to tell us?
I feel better than ever being a mycologist and part of this society, and because of great people like you too.
Hey, we're glad to have
That's
you.
terrific.
Nice plug. Thank you.